Today, I will be speaking with Dr. Roland Chapurlat, who is the senior author of the article titled Plasma Cartilage Acidic Protein 1, measured by ELISA, is associated with the progression of total joint replacement in postmenopausal women. Roland, I want to thank you for taking the time to speak to me and our listeners on behalf of the authors of your paper of this important observation. So let's start, okay? Could you please explain what cartilage acidic protein 1 is, which cells express it, and what the soluble form of it has been associated with?
So, CRTAC1 is a glycosylated protein that is a matrix protein that's found in the cartilage. And so it's in the relatively deep layers of the cartilage as well as the calcified layer of the cartilage. It's relatively specific for the joint, for the cartilage, but it's been found also in brain as far as I know. But essentially, this is a cartilage glycosylated protein. And so it can be found, it's expressed in synovial as well as chondrocytes, but not in bone cells. So it's not supposed to be in the subchondral bone, which is an important area of the pathophysiology of osteoarthritis. And so in prior studies, it's been found to be associated with the burden of osteoarthritis in at least 3 cohorts, one in the Netherlands, one in the UK Biobank, as well as a cohort in Iceland. And here the approach was to look for new markers, new biomarkers for osteoarthritis. And so they used proteomic approaches. And there were a number of proteins that were found to be associated with the burden of osteoarthritis, as well as the progression of osteoarthritis. So burden of knee, hip, spine, and hand OA, and the progression for knee OA. So, this was pretty convincing in terms of statistics. And however, of course, proteomic approaches are quite difficult and expensive, and they cannot be used clinically. And so we wanted to know whether a more simple approach assay would be feasible, and whether this would provide comparable results, which is why we have tried an ELISA that's commercially available, and looked at the association with OA and the progression of OA, which is I believe most important. And so we looked at the progression of OA in our cohort of postmenopausal women, and essentially we wanted to sort of calibrate the result of the association with another marker that we have already published, which is the well-known CTX2.
Okay, thank you. So that already answered my second question, really. And why did you pick postmenopausal women rather than a more diverse cohort?
Well, in fact, this is a cohort we have that's in our hand well characterized, which is a cohort that was recruited something like 30 years ago, the Offaly cohort. And so initially this cohort was designed to look at postmenopausal osteoporosis, which is why these are postmenopausal women. And we have over time collected also outcomes for and predictors for osteoarthritis, and specifically knee osteoarthritis, with several X-rays 4 years about to look at the progression. And we have also collected the outcome of joint replacement at the knee and the hip. And so in this analysis, we had an 18 years duration of follow-up. And so we were able to examine the relationship between the baseline measurement of markers and the outcome 18 years later. And there's also a sort of a biological rationale. That is, when you do a deletion in a mice model, a deletion of the gene of CRTAC1, there's a phenotype of osteoarthritis. And that's observable only in female animals, but not in male animals. So, but I believe that's probably to be, so our results are probably to be replicated in the cohort of men.
Sure. Sounds like a good, excellent reason to actually study it. Thank you for explaining that. And what other biomarkers were set? I'm sure you studied other ones.
So yes, so we have already published that in the same cohort measurement of CTX2. So CTX2 is a degradation, is a type 2 collagen marker of degradation. So we have found that higher levels of CTX2 are associated with more progression, and specifically the higher levels are also associated with a higher rate of joint replacement at the knee and hip. So in a prior paper, we have found that we have measured the CTX2 in the wall cohort, and we are able to compare those with joint replacement with controls. And in this present analysis with CR-TAC1, we've made a smaller analysis, which is a case control analysis. So we've measured CR-TAC1 in those who had joint replacement, matched with two controls. So match based on BMI and age. So probably less statistical power than in the prior analysis with CTX2, but anyway, we found that there was a comparable association of the two markers. So odds ratios around 1.7 per SD change, and which were also comparable to the prior analysis using proteomic approaches in the three other cohorts. So I think that's pretty compelling.
Good. So I think you did get the results of your study, told us that. And so really that's, to me, it sort of goes to the next question. Aside from we can measure it, and the rationale was very well explained of the cartilage acidic protein 1, are there any advantages, disadvantages over other biomarkers that you believe? In particular, the cetalopeptide, which most people are much more familiar with, and so probably not easier, but
yeah. As you know, we don't have many markers of osteoarthritis and specifically predicting the outcome of joint width narrowing or joint replacement. CTX2 has been shown to be able to do this job. However, this is a urinary marker, and so the variability, intra and individual variability is not very good. Sensitivity is not perfect as well. And so we are clearly in need of other markers, and specifically if they can be serum or plasma markers, because generally the reproducibility of these markers is better. And what we also found in our analysis is that when we entered both markers into the model, they were equally predictive. So suggesting that they were predicting different aspects of the biology of osteoarthritis. And so that maybe they can be combined and not being exclusive, if you wish. But I believe that blood markers, plasma or serum markers are probably easier to measure, to obtain, and more reproducible. That's probably the long-term goal.
Right. And so now, what would you recommend people should measure if they're looking for the risk assessment for their patients? Well, at least in postmenopausal women.
Mm-hmm. Well, I think that we probably have to make new analysis in larger cohorts, maybe older women. The mean age in our analysis at the baseline was 65, so probably that would be interesting to look at that in older women. Probably in one or two other populations to make sure that this is not a cohort effect. And certainly we would be also interested in men who are also affected by knee and hip OA. So we need some degree of replication of our results. And probably, well, one way we're evolving in the field of biomarkers nowadays is to combine several markers to build scores. And so that's probably a statistical approach to look at this.
Right. And is anything you want to add that we may have missed? I forgot to ask.
Well, I don't think so. I think
Okay.
you have
Good.
all the question.
Thank you. And
You're welcome.
I want to thank Dr. Roland Chapurlat for taking the time to review the findings of this paper and the implications. To our viewers and listeners, please read the complete paper. Plasma cartilage acidic protein 1 measured by ELISA is associated with progression to total total joint replacement in postmenopausal women. Sometimes my tongue and my brain are not in sync. It is available on the journal's website at jroom.org, and both will appear, and it will appear in the February print edition of the journal. I want to thank you for joining us and hope you are going to join us next month when I speak to the author of another paper of interest from the March edition of the journal. And I'd love to hear comments or any suggestions, which can be given to us on X, JRoom, or by email manuscripts@jroom.com. Thanks.